Hello, my name is Karen Nuñez and I'm a PhD student at the International Laboratory for Human Genome Research from the National Autonomous University in Mexico. The title of my work is Computational Modeling of the Regulatory Network Controlling Monocyte to Dendritic Cell Differentiation. Dendritic cells are the antigen-presenting cells that can be obtained from the stimulus of the monocyte with colony-stimulating factor 2 and interleukin-4. That stimulation leads to the activation of 4 signaling cascades: MAP kinase, P3K, JAK-STAT, and NFKB. The dendritic cells derived from the monocytes are the most accessible due to the easy access of monocytes from the bloodstream. Regarding that this process of differentiation is widely used, little is known about the regulatory network required for it. The aim of our work is to study and characterize the epigenetic mechanisms that control dendritic cell differentiation and integrate this information in a dynamic model to determine which pathways are possible therapeutic targets. The methodology that we follow was to set bases from the literature knowledge about the differentiation of the monocyte to the dendritic cell into a logical model, and we look into high-throughput data from ChIP-seq and RNA-seq to discover the new regulatory interactions between transcription factors and specific genes of dendritic cells. To our first model, we integrate the regulatory interactions that we found from the high-throughput data analysis to complete our model and perform simulation to corroborate our findings. The results from our high-throughput data analysis of the 6 histone marks of monocytes, dendritic cells, and macrophages as negative control of the differentiation. In the figure 3, panel A, using chrome-HMM, we identify 10 chromatin states that describes active and inactive regions with a combinatory of different histone marks. In the panel B is shown a genome browser screenshot of the region of the gene IR4. That is a transcription factor that participates in the dendritic cell differentiation from the monocytes. In the first row, the macrophages are shown. According with the color code from panel A, the gene is inactive. In the second row are the monocyte, and in the third row are the dendritic cells, and the dendritic cells is the only cell type where the gene is active. Using the regulatory regions from our previous analysis, we look for the transcription factors binding sites of the transcription factors related with dendritic cell differentiation. In figure 4, panel A, in the x-axis are the specific genes for dendritic cells, and in the y-axis are shown the regulatory transcription factors. The squares color represent that a site for a transcription factor was found. The asterisks represent a site that was also previously reported. We can highlight that IR4 has 13 new sites for specific dendritic cell genes. In the panel B, the sites discovered were from the regulatory interactions between transcription factors. In the x-axis, it shows the target transcription factor, and in the y-axis, the regulatory transcription factors. The colored square are the sites that we discovered, and in the asterisk are the previously described sites. Once we identify the regulatory interactions between transcription factors and transcription factor with the specific dendritic cell genes, we put it together into a logical model shown in figure 5, panel A. Then we perform several analyses in order to prove its fidelity with the biology. First, we look for the specific stable states, and we have 1 per the combinatory of the stimulus. In the figure 5, panel B, we have 4 stable states as a result from our model. In the first column, without any stimulus, the monocyte goes to cell death. The second row, only with interleukin-4 as inputs, we have the specific markers for the monocytes. In the third column, with colony-stimulating factor 2 as input, the macrophages can be obtained with their specific gene markers. In the last column, with colony-stimulating factor 2 and interleukin-4 as inputs, we obtain the dendritic cells derived from the monocyte with their specific gene markers. In order to test if the stable states can be reached through the time, we use the tool MABUS in order to test it, and we found in figure 6 that the trajectories of the probability to reach a specific cell type were obtained. In panel A with the interleukin-4 as input, in the orange curve we have the monocytes that decay, and in the green curve we have the specific markers per monocyte stimulated. In panel B, having the colony-stimulating factor 2 as a stimulus, we show in the orange curve the monocytes that decay, and in the blue curve, we have the specific markers that grows for the macrophages. In the panel C, with colony-stimulating factor 2 and interleukin-4, as a stimulus is shown the orange curve of the monocytes that decay, and in the purple curve with the specific markers of the dendritic cell derived from the monocytes. As conclusion, our strategy allows us to put it together a comprehensive regulatory network involved in the control of monocyte to dendritic cell differentiation. The result presented here casts light on the complex regulatory networks governing monocyte to dendritic cell differentiation. These findings can help improve the development of immune therapies based on dendritic cell usage. And I would like to thank my lab mates, my collaborators, and the technical support that I received to complete this work. Thank you all, and I'm ready to take your questions.